of the faculty that you'll be hearing from, I'm probably the one person who is a practitioner of stem cell biology. That is, I use it in the treatment of patients. And I thought it might be of some interest to try to think about how stem cells are being used today for the care of different kinds of diseases, and how they might ultimately be applied. And so I'm hopeful that we'll be able to get to some examples specifically of the way that we have gone from really basic experiments that have taught us that stem cells exist and what they can do, to how that actually translates into something that can be in the hands of a physician to ultimately result in a benefit for a person with a disease. So I know that you've had the introductory talks, but I don't know if you've had the perspective of a physician, which is basically that these are entities that are more than just of interest biologically. We all depend on them for life. And that if your stem cells decided to check out today, you would have a couple of weeks before you would not be able to survive. And the reason is that we all have tissues that turn over very rapidly. In fact, we have to make 10 billion new blood cells every day. And that's a tremendous chore that can only be sustained because we have stem cells. So stem cells that are a part of the skin, of the intestinal tract, and of the blood are absolutely essential for maintenance of life. And in their absence, there are people who have been through this in the context of what was chemical warfare in World War I and radiation warfare in World War II. And the effects of those, as you know, are devastating. So I just want to go back to some of the signature characteristics of a stem cell. I know you've had this, but review is something that I know you also know about as teachers. And the idea that stem cells have a very different set of characteristics from other cell types. Remembering that these cells are essentially inert. They conduct none of the business that's important in terms of the function of a tissue or an organ in our body. Yet they are the source of cells that can then go on to acquire all the mature, highly specialized features that can make up a cell type that might, for example, carry oxygen, as in the red blood cell, be a platelet that's important in blood clotting, or a white blood cell important in fighting infection, or any of the other more mature cells that make up the body. As I mentioned, these cells need to be produced at a level of 10 billion blood cells a day because some of them have very short half-lives. Some of the white blood cells that are critical for fighting off bacterial infection, for example, only last 7 hours in our bodies. And so they must be produced in great numbers. And yet you could imagine if they were dependent in a 1-to-1 or even 1-to-2 or 1-to-4 ratio with more immature cells, that the amount of stem cells you'd have to have would be enormous. So the good news is this system is one in which stem cells are relatively modest in number. They produce cells that go on to differentiate and expand dramatically, which then drive cells onto a fully differentiated mature phenotype. But along the way, the stem cell, when it creates one of these daughter cells, it can divide such that one of the other daughter cells is exactly identical and is a stem cell again. And so it essentially remains on the shelf for the next demand. And that property of being able to have this dual outcome is something that's quite distinctive about stem cells. Now, as you know, stem cells are not all created equal. They come from different times in our development. And I think you've heard already about embryonic stem cells that are derived from really the very early structures following fertilization of the egg. In the human, this all occurs around day 7 to day 10, yielding a structure that's about as big as the sharp end of a pin. And it's from this structure that you can derive the embryonic stem cell population that has gotten such attention because it can become all different cell types of the body. But by about 2 weeks of development, the cells that are capable of forming these pluripotent cells that have all this capacity, is generally lost. And cells end up having a much more restricted outcome in that they can become really only more cell types of the tissue from which they are derived. And they are so-called multipotent. For example, the blood stem cell is a classic example of this. It can make all of the different cell types of the blood, but it cannot make something like a heart cell or a brain cell. So it's very limited in what it can do. And again, I'll point out that this transition occurs very early in development. So you've probably heard about things like umbilical cord blood cells. They're very powerful cells, but they really function as adult stem cells. They are very restricted in what they can become. And again, just to reiterate this difference between the cell types, an embryonic stem cell can become all different cell types. It is so-called pluripotent, whereas limited potential in the adult stem cell, and it's multipotent. These are cells that can be grown in great numbers. They have a capacity to form tissues in such an unregulated way that if you were to put these cells in an animal, they would form a tumor. They form a so-called teratoma. And our methods to control them in terms of what they can become at this point still remain fairly poorly defined. The adult stem cell doesn't have this problem of becoming teratomas because it has this limited potential. The problem is these are relatively rare, and some of them are very difficult to isolate. If they only come from the tissue from which of the cell of interest. So for example, the blood stem cell we can get from the blood or bone marrow. But as you might imagine, if you're interested in a stem cell that's from the brain or the heart, you've got a very different challenge in terms of being able to identify these cells and isolate them. We know, for example, that the brain stem cells live very deep in the cortex of the brain, right in the center of the brain. And so you can imagine that being able to isolate those, it would be difficult to get volunteers to come forward. But these are the ones that we use clinically at the moment. Now, how did we experimentally first define that there were such things as adult stem cells? And I'll go through just the classic experiments. The hypothesis really came from observations in the field about the survival of animals and humans from exposure to radiation or exposure to agents like nitrogen mustard that was used in World War I. And it was found that in an interval following exposure, those individuals or those non-humans who were animal subjects would essentially lose their blood cell production, but it would come back. And the hypothesis was that this was because of a stem cell. So the way the experiment was done was by actually a combined effort of a radiation physicist and a biologist, Jim Till and Ernie McCullough at the University of Toronto back in the late 50s. And the way they did this was basically to say, okay, if I take bone marrow from one animal, and I now irradiate two animals, and I give one of them no bone marrow, and just see how it does, this animal will die, uniform fatality. But if I just inject the bone marrow into the other animal, if I inject it into the tail vein, this animal will recover. It does well. So it was a demonstration that there must be cells that have the capacity to regenerate the blood system that was missing in this animal. So how do you then go about really defining what are the features of a stem cell? That is, that you would need to define this ability to differentiate into multiple cell types and to self-renew. Remember those two signatures of a stem cell. So what Till and McCullough did was first they examined the tissues. When they did this transplant, they looked in places like the bone marrow where we know blood cells are made, and they found that there were blood cells being produced there. But curiously, when they looked at the spleen, they found that there were these lumps. Typically, the spleen is a very uniform appearing tissue. It has a kind of a sheen to it. And they found that there were lumps on the surface. And these lumps were something that they could actually take and isolate. And when they did this, they could actually count these, and they would isolate them. And they said, maybe this is the growth of blood cells coming from one stem cell that might have been transplanted. Might this be a way in which we can quantitate that an individual cell can contribute to all the different blood lineages, that multi-lineage differentiation, and possibly self-renew? When they saw the cells that went into the bone marrow, those were just admixed. It was just a sea of cells. But this, they said, maybe this represents a restricted site where one cell has lodged and is now growing up. This was a very critical step, because then they could say, okay, I'll take one of these areas, I'll isolate this, and I'll look at it under the microscope. And what they did is they found that indeed it had red blood cells, and it had this is an emerging red blood cell, as well as white blood cells of different lineages. So they said, well, if it's true and it came from a single cell, it certainly is becoming many cell types. So it's one of those two signature features of a stem cell. But they couldn't tell for sure that it came from one original cell. So they had to do something different, and they did a very clever thing. What they did is they decided that they would take this animal that was receiving the new bone marrow from a donor, and now they said, we know that if we give radiation, a lot of the cells will die, but we won't give as much as we did initially so that all the cells will die. We'll just give enough radiation so that we might induce some mutations. And we might be able to see these mutations if we now look at the chromosomes of the cells. And so they did this. And what they found is that in groups of cells, they could find cells, some of the cells within this group, for example, might be dividing, and that allowed them to do what's called a chromosome spread, and they could look at the chromosomes. And some of them were abnormal, so they'd find a fragment here, an additional arm there, and they'd say, okay, we found it in one cell that came from this group. Did it show up in others, and did they have the identical signature? And indeed they did. So they would count, and they counted hundreds and hundreds of these. And they counted hundreds of cells from an individual collection, and they found that within a given nodule, the same signature changes in the chromosomes could be found. So their reasoning was that they all had to come from the original same cell type, and that now all the descendants would bear this same abnormality. So that was the first demonstration on what's called a clonal level, that an individual cell could become all of these different blood cell types. So that showed that differentiation capacity. Now what they did is they said, okay, now we're going to take one of these and we're going to transplant it again. So they did what's called a secondary transplant. So this was their ability to demonstrate multipotent differentiation, one arm of what you need to define a stem cell, and the other was the self-renewal property, which they did by a rather complicated experiment. But basically, they took a bone marrow from donor A, for example. They injected it into a radiated recipient, so animal B gets its bone marrow destroyed by radiation. They then allowed the stem cells from donor A to engraft. So now this individual animal, though it's still B, has the blood of A. Just like if I was to donate my bone marrow to one of you, you would now have blood that came from me. Now what they did is they said, okay, now we're going to take the bone marrow from this guy, and we're going to transplant it into another recipient. And what they did, in fact, was not take bone marrow, but they actually took one of those spleen units, those colonies. And they found that, in fact, that was capable of engrafting and fully reconstituting another animal. So essentially what they're saying is, so I know a stem cell went into that spleen, it made a nodule that made all those different blood elements. Then I know that I could take that same group of cells and transplant it, and it could function as a stem cell again because it could make, again, all of the blood necessary to have this animal survive. So they demonstrated for the first time that there was such a thing as an adult stem cell. And this is really actually the gold standard for how you define an adult stem cell type. So this experiment, first published in 1963, was the first definition of adult stem cells. Now, as you might imagine, that led to a tremendous boon in our understanding of this cell type and in thinking about the blood system as a great model for how tissues are organized generally and how stem cells participate in tissue organization. So I'll tell you about some of the things that we learned. The first is that the blood system had been characterized really since the early days of cell biology. Von Leeuwenhoek, in fact, the first cell types that he looked at that were not from plants were actually blood cells. He looked at the blood cells of a salmon. So the first microscope ever was one that looked at blood cells. And he could say that these were corpuscles, as he called them. And from that early day, morphology of the blood was an area of fascination. And it was really one of the early, also the ways in which cell biology was first defined. And the hypothesis of cell biology is, of course, that all cells come from other cells. That cell theory is one that is played out in the blood, in people looking back at where blood cells came from, the bone marrow, and they could find that actually now cells all started to look pretty similar. The color scheme was not agreed upon at that time. And they said, well, okay, these are all looking pretty much the same, but they make different cell types. What's going on? So what has happened since Tilden McCullough first defined that a stem cell existed, was they said, okay, now we really want to be able to isolate them, not just say that they exist by virtue of what they did, but can we go back and identify them prospectively? Can we pull them out of the sea of this otherwise similar looking cells? And so what people started doing was basically saying, okay, I'm going to take some of these cells and I'm going to inject them into a rabbit, for example, and raise antibodies. It was really the development of antibody-based technology that allowed people to be able to start to sort out what cells could do. And the way they would do this is basically say, okay, here we have something that we know recognizes a subgroup of this pool of cells in the bone marrow. Now let's do what Till and McCullough did and transplant it. Does it create all the blood cells or not? And from those kinds of experiments, people said, oh no, some of them only make cells that make up What is the common myeloid series? The myeloid series are things like erythrocytes, platelets, granulocytes, but it didn't make any of the lymphocyte series. So I said, that's interesting. You can subset a group there. You can subset a group that only makes lymphocytes. You can subset a group that makes both of them, but only lasts for a short period of time, and some that last for a very long period of time that are truly stem cells. And so about a 30-year interval of experimentation went forward, and we were able to isolate these subgroups. Now that was very informative because it allowed us to start to understand how this tissue, the blood system, is organized. And this has become the paradigm for the way most tissues are organized in adult animals of virtually all types. And that is that indeed there is this progression from a population of cells that are stem cells to a progenitor to a precursor to a mature cell. Now that occurs with a gradual increase in the differentiation state, the status of the cells. That's something that then has allowed us to be able to say, okay, we can both define these by their properties of being common lymphoid cells or common myeloid cells, and we can tell their difference by virtue of things that are on their surface, proteins that we can identify with antibodies, so we can use antibody-based methods to isolate them. We can isolate them. That means we can study them molecularly, so we could actually discover what it is that allows a cell that's a common multipotent progenitor that becomes just a lymphoid-restricted progenitor. And those kinds of molecular experiments have all descended from that early experiment by Till and McCullough. And that's led to a very complex map that tells us that there are these different subsets of cells. They have different molecules that help determine what they are. Their molecular signature is very defined now, as well as certain proteins that we know control them. So some of these proteins actually are capable of generating a robust production of a particular cell type. That one of those proteins is called granulocyte colony stimulating factor, based on some of the crude early experiments that were done just in the laboratory in a petri dish. But that actually has turned into one of the most powerful therapies to protect people who are getting cancer chemotherapy. So this is also known as Neupogen. It's used every day by literally tens to hundreds of thousands of people who are getting cancer treatment because it allows them to get cancer therapy that would otherwise suppress their immune system. They take this drug, their blood cell production of this particular subset of cells goes way up, and they no longer have problems with bacterial infections. So this information has led to both molecular understanding, biologic understanding that's actually translated into drugs. Okay. Well, that's one feature, the ability to identify what characterizes the differentiation status of a cell. But one thing that was also found is because we could now take subpopulations, isolate them, and do this transplantation assay that Till and McCullough did, we were also able to find out that self-renewal, that is this ability to repopulate a tissue, is very restricted. It doesn't happen when you get to these more mature cell types. It's only at the stem cell level. So cells have this reduced capacity to self-renew if they're anything other than a stem cell. That's important because it means that when we transplant, unless we have stem cells there, you won't get a durable engraftment. And if you just give someone blood transfusions, you can count on those cells only lasting a short time and the person ultimately dying. Now, one of the other things we found is that these cells are actually, at the point of this, at the stem cell level, don't divide very much. In fact, these 10 billion new blood cells that need to be made every day are largely made by the population of cells that's in the progenitor or precursor population. And that is reflected, oops. This is now what's called a transient amplifying pool. It's a population of cells that's rapidly amplifying up, but it only lasts a short time because once it's entered into this pathway of differentiation, it inexorably scoots down to a fully differentiated state. So in fact, the proliferation of things that are in this pool is on the order of about every 18 to 72 hours, a new cell will be made. But a stem cell in a non-human primate only turns over about once every 6 to 12 months. So our stem cells are very deeply quiescent. And they are very rare. So we only have stem cells that number in the millions, and yet they're producing these 1 to 10 billion blood cells a day. So they're a rare entity to try to find. Okay, so if we think about adult stem cells in the blood system, and now having the ability to potentially apply this to other tissue types, These are the general characteristics that have emerged. That indeed you can identify a stem cell biomolecular signature, that it is self-renewing, that it generally is slowly dividing, and that it's rare. Now, it's rare partly because of the fact that they can self-renew, so you don't need many. They'll have the ability to replenish themselves. Partly also because a self-renewing population that isn't very differentiated, if that got out of control, you could imagine that would be a problem. And that's a model for cancer that we'll talk about in a few minutes. In addition, they must be, the idea that they're slowly dividing is something that probably also has a good basis. And it may be because if you were exposed to a toxin, for example, if you had a rapidly dividing stem cell pool, it might be very sensitive to it. And yet these slowly dividing cells are accompanied not just by slow division, but also by very active pumps that exclude toxins. And so these cells generally are very resistant to cell death induced by different chemical toxins. Well, that's led to our not only understanding of the biology, but also their use clinically. And so I'll just give you a quick timeline about what's happened, because this is actually the 50th anniversary of the first use of stem cells in a human in the setting of a bone marrow transplant. And this setting was done 50 years ago in a very unusual context. And frankly, if we were to do those kinds of tests today, we would probably either end up in court or certainly end up being banished from the scientific community, because these early experiments were ones that were generally devastating in terms of their impact. And it was really only after an interval of over 20 years that it became something that became more than just a rare curiosity in an academic center. So the maturity of stem cell biology going to stem cell therapy is one that's measured in decades. I think that's something we could also expect for embryonic stem cells. Today, we now do tens of thousands of transplants, and they save lives very dramatically. So for example, in the setting of autotransplant, that means that the person serves as their own stem cell donor. This is used in patients who have things like relapsed Hodgkin's disease or other types of lymphomas. In this setting, people have relapsed Hodgkin's disease. It used to be that they had a mortality that was essentially 100%. If you use this approach, you can now cure about 80% of them. So it's had a dramatic impact on people's well-being. And I have to say, one of the reasons I went into this field is because I was feeling sorry for myself as an intern until I bumped into somebody who was actually waiting on me in a store, and I was moaning that I was taking care of all these very sick people who were on the transplant unit. He said, "Oh, I had one of those." And he was a young guy who had basically been rescued from certain death by virtue of having undergone a bone marrow transplant. So this is a therapy that has demonstrated that stem cells, even if viewed in this narrow window, can have tremendous impact on people's well-being. And these have generally been used in young people. So the person I saw was actually in his early 20s. And it's very common that it's those people who need this approach and who can actually be benefited by it. So of course, this model, which is the one that I really came to when I was studying stem cells early on, is one that offers tremendous hope for the idea that we might be able to go beyond just the blood system and take advantage of some of the other ways in which we can either modify blood stem cells or use stem cells that might come from other sources and have an impact on diseases other than just diseases of the blood system. So how are we thinking about using stem cells broadly? Now that there has been this explosion of our understanding of both adult stem cells and embryonic stem cells, what are the ways in which that will ultimately translate into an impact for you and me? And one of those, the simplest way to think about it, is as a cell therapy, as essentially a replacement part. That's basically what we do with bone marrow transplant. We give patients drugs that wipe out their bone marrow, and we replace it. But there are many other ways in which I think this field will have an impact, and I want to just update you on this so you have a sense of it, and also how stem cell biology can have an impact on thinking about diseases. So one of them is the idea of making new drugs. The other is that it will influence our way of thinking about cancer. And finally, the idea of being able to generate new models for disease that can, again, also help with drug development. Let me first focus on the idea of cell therapy. As I mentioned to you, we are generally using it as a way to replace injured tissue in things like bone marrow transplant. But there are other examples of this. One of the reasons why you can give someone a skin graft after a severe burn or a bone graft in the setting of orthopedic injury is because there are stem cells in those grafts. And those stem cells allow for replacement of the tissue. They then become incorporated into the tissue and are sustained. So they are a durable transplant, and that's a very important way in which stem cells are being used. There's an alternative strategy, which I'll mention shortly, about the way using the cells in a slightly different mode. But this idea is one that, of course, we'd like to apply now not just to adult stem cells, but to embryonic stem cells. And I'll just make mention, I'm not sure how much you've heard about embryonic stem cells. A little bit? Okay. Well, basically, human embryonic stem cells can be grown very abundantly in a laboratory dish. When they become a specialized cell type, they do this with a process that we understand relatively poorly. And the number of steps that's involved in any particular kind of specialization is something that's only starting to be sorted out. I think you probably heard about efforts to try to make them become insulin-producing cells from the lecture you heard previously. And one of the problems will be being able to get isolated subpopulations that do the interesting things that we're all excited about, like when you're in the lab late at night and you see a colony start to pulse, you both wakes you up a little bit, but you also recognize that some of these cells are becoming things like heart muscle cells. And what's interesting about it is that they can become this cell type, but also what's disturbing about it is that what might be nearby is kidney or skin or heart or muscle or skin or some other tissue type. And so one of the questions is, how do you get them to be very restricted in what they are, or how do you isolate them away from what's an admixture of cells? And so one thing that can be done is you can put in tags, and you might have also heard about this, experimental strategy, to basically allow you to pull out subsets of cells. And one of the questions is, well, how do you investigate what they do? For example, we've been interested in identifying cells that can become blood vessels. Well, they have all the characteristics in a dish, but to ultimately know how they'll work, you need to put them into a mammalian system. And I don't know if you've heard about this, but this is a commonly done strategy now, which is to take the cells and transplant them. We know, for example, that there are immune-deficient mice that can be bred in the laboratory that are so deficient in their immune system that if you put human cells into them, they don't reject, they engraft. So we can make mice that actually have a human blood system easily. And now that's been possible with other cell types as well. So I'll show you one example of doing this, where we took these cells that were lying down in the dish and looking as if they might become vascular cells. We then disrupted them so they were individual cell types. We had them in a mixture that was kind of a slurry. And then we put them into an animal. And only the human cells were green. The mouse cells were not. And they started to organize, actually. They went in as individual cells. They started to actually make these linear structures. And eventually they ended up looking fairly interesting. And I'll just show you. We put a dye in the veins of this animal. And in fact, they had become real vascular tubes. And what's interesting about this is, of course, we gave no instruction. These cells were able to, though, form these cylindrical structures. And not only that, they found a way to incorporate themselves into the vasculature of the animal. So they became part of the circulatory system. Really quite an extraordinary feat of self-engineering, if you will. It gives great hope that maybe we'll be able to do similar kinds of things in a human, that we might be able to actually just give cells and not necessarily have to really recreate the whole tissue in order for the cells to organize, self-assemble, and potentially contribute to something meaningful. So that's one way in which embryonic stem cells have the potential of making a difference as a cell-based therapy. Clearly, you've heard about the idea that maybe they could also be something that produces insulin in the setting of diabetes. Now, there's another way in which cell therapy is being tested today, and that is that the cells may not be incorporated into the tissue, but may actually be production vehicles, that they're actually little factories of proteins, very complex factories of proteins, but nonetheless that they don't need to necessarily become a part of the tissue. So, for example, this has been done in heart disease, where you try to take a cell and ask it to become a heart muscle cell. In fact, what it does is just elaborate sufficient good humors that it can actually be protective. And that experiment was done with what are called mesenchymal stem cells. This is a skeletal stem cell that's been shown to become many different types of mesenchymal cells, cells that really make up the sinew, if you will, of our body, of bone and cartilage and muscle and tendon and, unfortunately, fat. And these cells had been thought, well, maybe if you put them into a place, they would reprogram to become a heart muscle cell in a setting of need. And so experiments were done where they were cultured and then they were injected intravenously or into the cardiac tissue itself in the setting of a heart attack, a model of a heart attack. And what happened was they saw that there were changes in the function of the injured heart. This was done in a couple of different model systems, but in fact it didn't show that the cells that had been transplanted actually contributed. They didn't become a part of that heart muscle. They were providing something at a distance. They provided something that protected the cell types, but for reasons that are at this point unknown. And frankly, it's a very interesting way of using a cell-based therapy that has now moved into clinical application in humans. So you may hear of this. It's done in Europe fairly frequently, where people who have a heart attack actually will get a cell-based therapy, a stem cell, that'll be injected into their bodies. It doesn't end up sticking around, but there are at least some reports that it might have an impact on the function of the heart. Okay, now what about the idea of being able to think about the cells not just themselves as the unit of therapy, not just a transplant, but what about the idea that maybe there are stem cells in our tissues already, and they do very well at being able to replace the skin and the intestinal lining, where they're very good about making all those new blood cells a day. But in other tissues, they don't work so well. Things like in the brain, there are stem cells, but they don't repair after a stroke. Are there ways in which we can encourage them to be more robust in regeneration by using medicines? Can we use drugs to target them? So that's a strategy that's actually starting to have some experimental demonstration that is quite encouraging. And I'll give you one example of that. So first of all, let me say that as we've tried to understand what adult tissues have stem cells, there are now a number of them where it's actually been fairly rigorously demonstrated. So those include things like the blood, but also the brain. And certainly when I was in training, the brain was thought that you received your complement of cells in the brain in adolescence, and it was all downhill from there. And you did your best in college to get rid of some of them, and then, but you're actually all endowed with brain stem cells. So we can actually continue to generate new gray matter. And the question is, how do we do that better? The skin clearly has stem cells. They've been found in the eye, in the breast, in the lung, a number of different tissue types. I've already mentioned the ones that make up the skeletal system. They're actively used in clinical studies. Well, are there ways in which we can approach these? And one of them is to actually try to go after the place where they live. So I'll give an example again of the blood, partly because it's my area of expertise, but also because it's one that's been a great paradigm. So we know blood is made in bone marrow. Bone marrow is in bone. And so we've done a number of studies looking at what bone contributes to bone marrow. And what we found was that blood stem cells actually live in close proximity to the bone-forming cells, the osteoblasts, that lie on the surface of bone. And that led to several different possibilities. First, it actually led to us being able to visualize a stem cell in the body, an individual stem cell in a living animal for the first time. So I just wanted to show you this because I think it's a neat picture. But basically, for the first time, we were able to look into the bone marrow of a living animal, and we could see individual stem cells labeled white here. And they resided close to bone-forming cells, these green osteoblasts, immediately adjacent to blood vessels, red, and bone itself in blue. We could follow these sequentially in this animal. We would actually give it a bit of a glass or plexiglass skull cap and come back and look through its skull every day, and could see that some of the stem cells actually started to divide and make more than one. In any event, what we learned from this is that the stem cells aren't entities unto themselves. They don't live alone. They live in a community. That neighborhood, that microenvironment, that so-called niche, may be important in how the stem cells behave. We know that's true, and now we've been able to test it experimentally. And the way you can test it is basically trying to modify the activity of things like the osteoblast. And you can do that genetically, which is what we initially did. But in fact, there are also drugs that have been developed for the treatment of people with osteoporosis that target this cell type. And so one of our hypotheses was that if we understand sufficiently where an adult stem cell lives, and we understand what environment usually regulates its activity, maybe we can identify new compounds that might modify that activity to increase stem cell function. And we showed that now using a drug that's, again, in clinical practice for the treatment of osteoporosis for a very different indication to increase blood stem cells. And it's true it works in animal models. So we tested it actually in an animal model that mimics something that goes on for patients all the time, which is bone marrow transplant. And here what we did is we gave animals a limiting number of stem cells, and this happens clinically. And their mortality was very high. In this case, it was 72%. And things like umbilical cord blood transplant, which has a lot of very good blood stem cells, but not enough for a fully grown adult, the mortality is actually even higher than this. If we gave the animals, however, some of this drug, this PTH analog, the mortality went to essentially 0. And what happened in the bone marrow, this purple part here, we're looking through a slice of bone, all this purple part is blood cells being produced. There's a lot more vacancy if the animal just got the stem cells alone. So it looks as if, in that setting, we could actually improve the outcome from something that was a model of a human setting. So this is now entered into a clinical trial. And it's being tested around the nation, actually, in 8 different transplant centers for people who have a stem cell disease, things like an acute leukemia or aplastic anemia. For those individuals, you need to get rid of their stem cells, or their stem cells have already died. And you need to replace it. You need to replace them with stem cells from someone else. Now, typically, identifying a donor is very complicated. It's wonderful if you come from a big family because you have a greater incidence of being able to find a match. But for the rest of us, it can be very challenging. And depending on your ethnic and racial background, it can be even more challenging because people who register for these bone marrow donor databases actually tend to not be in a uniform mixture across our population. So a large fraction of people who need a transplant as a life-saving measure can't get one. They can't find a donor. And one ready source is actually umbilical cord blood, which is now being stored from delivery suites around the country and is actually a much better representation of the ethnic and racial mix of our population. So that's a very big resource. The problem is they don't have enough stem cells for anyone over 100 pounds. So you can start to use combinations of multiple different cord bloods. That's reduce the mortality. But one of the questions is, can we actually do better, make the stem cells more efficient by treating the microenvironment like we did here and like we did for those animals with using a drug? So that's actually now being tested in a multi-center trial. So it's one way in which stem cell biology has taught us a different approach to the use of stem cells, to using a drug as a way to modify stem cell function. Now, if you can apply this to other tissue types, this would, of course, be enormously powerful and something we're in the process of trying to do. All right, what about the idea of cancer? The idea that stem cell biology is just restricted to repairing injured parts is something that is a popular notion. But in fact, it's had perhaps one of its greatest impacts is on the understanding of cancer. And if we think about the blood system, where I told you there was this organization of a rare stem cell population that yielded descendant cells, some of which were very short-lived, if that applies not just to normal tissues, but also to cancerous tissues, then maybe we can think about cancer differently. The traditional model is thinking about cancer as being a group of cells that might be slightly different from one another, but are all equivalently nasty. But if you apply the stem cell model, then you might hypothesize that there's a particular subset of cells that may be a rare cell within a cancer that is capable of self-renewing, of generating more of itself, and in that way potentially being a cell that can lead to metastases, and yet can also yield the bulk of a tumor type that might be other descendant cells, and that maybe some of these cells wouldn't even have the capacity to stick around. Maybe like blood cells, they would actually die off fairly readily, and that the reason you can have a cancer stick around for so long is because of the cancer stem cell itself. And that model would have broad implications for cancer drug development. Because if you think about a cancer as a uniform entity of cells, then the way we develop drugs now makes sense, which is basically to just see which drug has the greatest impact on the bulk of a tumor. How much does it shrink in 2 dimensions? But if the stem cell hypothesis is true, then you might imagine that if you see a lot of cells get better from a therapy, if they're the cells that are just these descendants of a stem cell that wouldn't otherwise stick around, that's not particularly helpful because the stem cell has the capacity to regenerate the entire tumor. Maybe that's why we have cancer recurrence. We've developed an awful lot of drugs that have the capability of providing a remission, but a transient one. So this hypothesis is one that is very appealing in terms of thinking about how we might develop drugs, because if we can identify cancer stem cells, then maybe these cells would be the ones that we could target specifically and be able to eliminate cancer. So that model is one that had appeal and was tossed around as a theoretic notion for some time, but it's been recently tested. And the way it was tested was, again, going back to those early models that were used by Till and McCullough to first define a normal adult stem cell. And what was done was to actually take the normal blood cell system. As you remember, basically they could identify that a subset of cells was capable of engrafting an irradiated animal, and that those cells could yield all of the different cell types that made up the normal tissue of blood. Well, using the same model, but now identifying, and using some of the markers even, that identified the normal stem cell, what John Dick's group did, again up at the University of Toronto, was to say, okay, I'm going to take a leukemic population. I'm going to see if I can identify some groups of cells that when transplanted into an animal were capable of yielding a leukemia, where other groups of cells might not. And that's indeed what they found. So they did the experiment, and they showed that indeed they could take a subpopulation of cells, that those cell populations were able to yield the full spectrum of cell types that made up the leukemia. They then also did the self-renewal experiment of basically taking one animal that was engrafted and now use it as a donor to transplant another animal. So they validated those two core principles of what makes up a stem cell, differentiation and self-renewal. And that vetted the idea that indeed cancer does have cancer stem cells within it. Now that's been applied. I won't go through the details here for brevity's sake, but it actually does seem to be the case not just for the rare blood cancer, but actually for other cancer types as well. And indeed, there are at least now 6 human tumors for which this has been validated. There are a whole host of them that are now being reported in varying forms, and it looks like this will be a common paradigm. Probably not for all cancers, but for many, this model will hold true. And so that makes a difference in terms of the way we think about drug development, as I mentioned before. It also makes a difference in terms of, does that mean that the stem cell is actually the bad actor, that it's really the stem cell that causes cancer? That's one of the hypotheses, that maybe because a stem cell can self-renew, maybe it's the cell type that accumulates genetic injury. For example, if a mature blood cell gets a genetic mutation, it doesn't really matter. It's only going to last for a matter of days to weeks to maybe months. But if a stem cell is durable over the decades to, we hope, a century of our lives, they could be the ones that really accumulate all that genetic damage. And they could then yield leukemia. And so it is a hypothesis that stem cells, while critical to the function of normal tissues, may be very, very limited in number, partly to protect us from this event. But it's also true that you can take now, because we could identify subpopulations of cells, we could actually introduce genetic changes. So these subpopulations could be isolated in a petri dish and exposed to a genetic change that was associated with a human cancer, for example, a human leukemia in this case. And what was shown is that it wasn't just the stem cell that was capable of causing leukemia, that it could actually occur from a more mature cell type as well. So while the efficiency with which this cell type was able to yield leukemia was very high compared to these, it's not necessarily the case that all cancers come from stem cells. There are stem cells or stem-like cells in cancer, but it may be that there are more mature cells that have actually gone back and acquired stem cell features, if that makes sense. Okay. So the idea of thinking about cancer from the stem cell perspective, I think will make a difference in developing new therapies for cancer. One other thing that I very briefly mentioned is this idea that maybe we can use stem cells as a way to get a handle on a number of complex degenerative diseases for which we currently have very little therapy. So there are some diseases that have very clear specific genetic abnormalities. Sickle cell disease, very well known. But something complicated like diabetes can have many different genes that contribute. Many diseases are of that nature, and it's very hard to understand them. You can't really create an animal model that has all those complex traits that may be a part of it with any high frequency. And so we've been stuck. How do we study diseases that are very complex, particularly of things like diseases that affect the brain, where we can't go sampling people's brain to try to grow neurons in a dish? Might it be possible to actually create disease-specific embryonic stem cells from which we could actually grow the cell type of interest and try to really create the disease in a dish? So that's one of the things that drives the idea of somatic cell nuclear transfer, which is also called cloning. Now, this process then is basically to take an unfertilized egg, to remove the nucleus, and that's done with a simple pipetting technique. So it now becomes just a bag of cytoplasm, essentially. Into that, you can introduce the nucleus from another cell type, including a mature cell type. Now, this has been done in animals. This is actually how Dolly was generated. That goes on to actually tell this nucleus that it's no longer something like a skin cell or, in the case of Dolly, a mammary gland cell from the udder. That actually reprograms, and it becomes something that has the capacity to actually look like a new forming organism, a blastocyst, from which embryonic stem cells could potentially be derived. Now, I say patient. This has only been done in animals, not done in humans. This is the theoretic basis for and justification, rationale for wanting to do somatic cell nuclear transfer. Because if you could create embryonic stem cells from someone who you know has a disease, you might be able to then now go on to grow the tissue. So if they have something like ALS, Lou Gehrig's disease, we know you can grow those neurons. You might be able to grow them in abundance in a laboratory dish and actually say, okay, what is it that's distinctive about those neurons than, say, their relative who doesn't have this disease? And are there drugs that can forestall some of the changes that might occur? So the idea that this could allow us to create true models of complex disease is one very powerful reason why we were very interested in doing this. In addition, the idea that these cells, if we wanted to, might be able to be transplanted back into the original donor because they would not be immunologically foreign is also an appealing concept. Well, this notion of reprogramming is one that's very potent and very highly desirable from our perspective in terms of being able to develop new therapies, but it's also technically incredibly difficult. Even just getting people to be willing to donate eggs is a very big challenge. And so this has never been done in humans, and I don't know if it ever will be, frankly. But with this strategy, you could theoretically create a model of a disease in a dish. Now, another way that this might be possible is if you could use something other than an unfertilized egg. We know there are eggs that either fertilize or fail to fertilize that are in vitro fertility clinics all the time that get discarded. And so one of the ideas is maybe you could use a fertilized egg. And that also now has been shown in an animal model and work by Kevin Egan here at Harvard to actually say, well, maybe we could actually use this cell type as a way to try to get reprogramming to occur. And he's again shown that for an animal model, but whether or not that can be done for a human is unknown. Finally, there's one very important new technology that I'll mention to you, which was shown by a really revolutionary experiment that was done by investigators in Japan, where they took the skin cell from an adult mouse, and they actually introduced 4 genes to induce what's called direct reprogramming. They were able to put 4 genes into an adult cell. It reprogrammed the cell. It essentially rewound the history of the cell, so it forgot completely that it was a skin cell. It went all the way back to becoming a bona fide embryonic stem cell and could actually contribute to the generation of all tissues of an animal. So if this could be done in a human system, you could imagine the power of that we might all be able to have adult cells that essentially were our own individual embryonic stem cells that would allow us to either have a resource if the cells became useful for transplant, or certainly become a resource in terms of understanding some of the diseases that we may encounter. So this is an ongoing area of investigation that's very, very hot at the moment, and is something that I think we're all very excited about as a way to really understand how to essentially rewind the history of cells. So I've tried to tell you then about a number of different strategies to create models of disease in a dish, but basically all of these are ways in which stem cells ultimately might have an impact in medicine. And while we're currently using cells in the cell therapy replacement part mode, and that's generally the topic that brings the greatest interest from the lay press, it's actually really a very narrow way to think about stem cell biology, and that it can have an impact in the generation of lots of other kinds of treatments, and can actually have what were essentially unintended consequences in even our way of thinking about other diseases. So I hope I've been able to give you a little bit of insight into the way stem cells work, but also into the way we might be able to actually exploit that information to develop new treatments for people. And I hope that you'll be able to take some of this back to your students. So thanks very much. Yes?
You had the spleen cells with the mouse, and you were treating the lumps. And you took out the lump, and then you injected it into the mouse, and then you got the vascular system. Did you just inject that in the blood, or did you inject it into tissue, or whoever did it? Where did they inject it?
It was actually into the brain. Yeah. It's an unusual place to put the cells, but it actually is based on some of the prior systems that have been developed to be able to visualize and repeatedly go back to the same landmarks on the skull. It's a little hard to do that on the skin and the rest of the body, but you can easily see through the skull, and you can actually create these windows through which you can look repeatedly. Yes?
The 4 genes that they did there in the last, the 4 genes they had, what do they do normally? I assume
Right.
Well, it wasn't random, the ones they put in there.
Yes, you're right. And it was actually hard to predict which genes would work, and so people thought this would have to be a very big experiment. And who knew what combination it would take and how many it would take, and only the egg cytoplasm had the magic. And so the question was, could you really dissect out the biochemistry of the egg cytoplasm, which was a tack some people were taking. But this group in Japan just started to create a matrix, and they said, well, we think these may be candidates. And they started testing them individually and then in combination, and they hit upon this one combination. It was an extraordinary stroke of good fortune, but also dedicated hard work. The genes that they chose, some of them are known to be important in terms of they're essentially all transcription factors, so they regulate the expression of particular genes. Some of them are things like MYC are also known to be associated with malignancy development. And indeed, these cells do have the capacity to form tumors. So this idea is one that has tremendous power, but clearly has lots of details to be worked out. And whether or not you can now substitute something like small molecules for these genes. They had to introduce these genes, they're permanently on. If you might be able to say, okay, let's have the same effect, but now conducted by a molecule that we can place on the surface. When the cell is done reprogramming, remove it from the medium, that would be a tremendous advance, but that'll take who knows how long. Yes?
Blood stem cells.
Yes.
They, in the marrow, or they circulate through blood?
Yes, this is a very important question. So stem cells in the blood were first identified as being in the bone marrow, where new blood cells are made. But it was recognized that they couldn't have started there because during development, there's no bone. It really only happens that you form bone into the second trimester. So blood cells were being produced elsewhere, and there are at least 4 different places where blood cells are first being made. They only get to the bone marrow through the bloodstream. So it's thought that at least early on they do circulate, and then someone decided to look in the bloodstream of an adult animal, and they found that they were there in low abundance. And if they gave that drug that I mentioned that's important for raising white blood cells, Neupogen, it actually increases not just the white blood cell count, but the release of stem cells from the bone marrow into the blood. So it used to be, when I first started in this field, we would do bone marrow transplants, harvesting bone marrow, by having people go to the operating room, and we'd spend hours putting needles into the pelvis, literally by the many dozen. And now people go to the blood bank, and they sit in a chair, and it's like donating blood. It goes on for a number of hours, but they circulate. And it's actually an interesting question as to whether or not other stem cell types circulate. They clearly migrate during development, and one of the hypotheses is that maybe adult stem cell types may also migrate. They tend to be found very close to blood vessels. So it's still being worked out experimentally. Yes?
Did you happen to know if prostate cancer was a stem cell?
Well, so there is a researcher out at UCLA who just actually presented at our symposium on Friday, and it looks like they have defined a prostate cancer stem cell. Rituxan, absolutely. Yes, so it's an antibody-based therapy. So it takes advantage of the antibodies that were originally used to identify what's on the surface of different kinds of blood cells and blood precursor cells. And it was found that on a particular kind of lymphocyte, the B cell, CD20 was present. So it was used as a marker. Then what they decided to do was say, well, maybe we can use it as a drug for B cell tumors. And so many adult lymphomas are of B cell type and have this marker on the surface. Rituxan was tried, and now it's one of the most potent drugs we have for the treatment of that disease. So it works very effectively. It's a great therapy. It doesn't seem to have an effect on stem cells, but lymphoid cells are very unique in the sense that they may actually have their own stem cells. Yes?
Going back to the mouse, when those stem cells were injected, would they have set up that vascularization without an injury being there? Or is there
We
any
don't know
detail?
the answer to that.
Yeah.
Yeah. I mean, they are injected, so it's done with a needle, so the amount of disruption is pretty minimal,
Right.
but it's still real. Yeah. I guess you could wait for some healing to occur and then add them and see if time of healing matters. I don't know. But these were all done just with a crude injection. Yes?
Are they looking into heart damage as a result of cardiac, whether or not the stem cells could be
Used in that setting.
area that is no longer functional?
Right. So one of the ideas is that it might be possible to use embryonic stem cells to possibly create cells that could be in some way incorporated into the areas of injury. And that's actually been tested in animal models. That's a very complicated process, as you might imagine, because in addition to being a pump, the heart is a very complex electrical circuitry. And disruption of that circuitry can be fatal. So when trying to do those kinds of studies in a human, you have to be absolutely sure you're getting it right so you don't end up disrupting the feedback loops that are important in maintaining the electrical conduction. So some studies have been encouraging in that regard, but I think it's still early on. The other that I mentioned to you, though, is this idea of using these mesenchymal stem cells as a way to essentially try to prevent injury, to try to not so much incorporate in and not become new muscle, but actually to provide something locally to the environment of injury. That is proceeding with lots of clinical studies at the moment. There's sufficient activity there that it's being done fairly routinely in some countries. I think it's still something we don't feel comfortable that we know enough about, but will ultimately, I suspect, turn into a more common way of approaching this injury. It doesn't help people who already have severe congestive heart failure, and that really is, at this point, still a goal that is unrealized. Thank you.